Our next speaker is Vinicius Adriano Vieira. He completed his medical degree at the University of Sao Paulo, and he did all his medical training in Brazil. In 2018, he joined Dr. Golder's group at Oxford, and he's still a PhD student, believe it or not. And he coordinates the court, the Durban court, and he's working now, and he's going to show us his research with a group of a pediatric group that controls the virus. That's it.
Yeah. Thank you. Hello. So first, I want to apologize, but I think I'm afraid I cannot speak in Spanish. I want to thank you, the organizing committee, for inviting me to give this talk. I'm not sure how familiar the audience is with pediatric HIV infection, so I want to spend the first part of the talk trying to explain why these elite controllers in pediatric population are so rare and how they might be a good model for remission in some aspects. And then I want to share some preliminary data of the work I started as part of my studies in Oxford, working with Professor Philip Gouda, who I am representing here now. So here is a graph showing plasma viral load versus years of viremia. So as you know, the majority of the individuals rebound soon after treatment interruption, but it has been described a few cases of individuals who are able to maintain undetectable viral load for years after treatment interruption, like the Mississippi child, some adult cohorts, and also more recently the South African child. And these individuals, they achieved remission, and what they have in common, they usually start therapy in a quite early stage of infection, but the mechanism that leads to remission is not completely understood. And on the other hand, we have the elite controllers, who are individuals who spontaneously control viremia, being treatment naive. And in adults, around 0.5 to 1% become elite controllers. But in children, if they exist, they are scarcely described in the literature. And to understand why they are so rare in this population, it's important to understand how the immune system develops and matures. So during early infancy, the immune system is very tolerogenic and is skewed towards a type 2 response, which is very bad to control intracellular pathogens like HIV. And along the years, the immune system becomes more aggressive and more of an adult-like response and is skewed towards a type 1 response. So infants usually have a very low production of type 1 polarizing cytokines like IL-2 and interferon gamma. And although you can detect some HIV-specific response, they are less polyfunctional and less frequent than the observed in adults. So children usually failure to control viral replication, and they progress faster to AIDS than adults, and the mortality is up to 50% by the age of 2 if treatment is not started. However, around 10% of these children achieved mild and late childhood, maintaining a normal CD4 count despite not being on therapy. So they are much more frequent than the adult non-progressors, and they share some similar immune phenotype to the SIV non-pathogenic infection, like low immune activation and exhaustion, especially on CD4 T cell compartment, and very low expression of CCR5 on long-lived memory cells. So although they have some HIV-specific response, they are not robust, and they failure to control viral replication, but maintaining a healthy CD4 count. And on the other hand, you have the elite controllers who not only maintain an average normal CD4 count, they also control viremia. And although they are quite a heterogeneous group, what is most evident is a very strong and rapid CD8-specific response. And the evidence for that is some temporal association between viral drop and T-cell-specific response. Also, some experiments depleting CD8 T cell leads to immediately viral rebound. And elite controllers also have a higher polyfunctionality and cytolytic profile compared to the other HIV-infected groups. And this population is particularly enriched with HLA-B57 and HLA-B27, which targets very conservative regions of the virus. So to be an adult-like elite controllers, the children would have to mount a response that is quite not compatible with the stage of development of their immune system. So that's probably explain why they are rarely found on the group of HIV-infected individuals, and only a few cases of case report has been described in literature in the last few decades. But early this year, we published a paper describing 11, and actually now we have 12 pediatric elite controllers coming from different parts of the globe. And we estimated a prevalence of 5 to 10-fold lower than adults. And it's striking of these 12 elite controllers, 11 are females. And it's well described in the literature, the sex dysmorphism on the immune system. So female usually upregulated more interferon-stimulated genes. They also have a stronger type 1 response and enhanced capability to produce antibody compared to males. Also, some important immune genes are located in the X chromosome, like the TLR7, which we know is not, the X gene is not completely inactivated. And also, estrogen is a more immune activator hormone, while testosterone is more immunosuppressive. So, for that reason, women usually are less susceptible to viral infection and they respond better to vaccines. They also, for HIV, achieve a lower viral set point, and they are 5 times more frequent elite controllers than males. However, because of this robust and immune response, they also have more immune activation, and they usually progress faster to AIDS by a certain time point. So another interesting find about this small cohort of elite controllers in pediatrics is the time to achieve a viremia. So in adults, usually they achieve viral control after acute infection, and most of them are controlling viremia by the end of the first year of infection. While the non-progressor child, although they don't control the virus, they only achieve the viral set point by the age of 5. Our elite controllers, they only achieve viral control by the age of 6.5, which is similar to the non-progressor and quite different from the adults. Another finding seems that the elite controllers in pediatrics seems to be short-lived. So the rate of loss of viremia is 1.2% per year in adults, and our elite controllers, 5 of the 12, has already lost viral control. And what we found is that the decline of the CD4 count during viremia control predicted those who would rebounded. So in red, we have those who are transient elite controllers, and in blue, those who are persistent elite controllers. And that was found only for absolute CD4, but not relative CD4 count. So to be pediatric elite controllers, they need to survive for years with ongoing viral replication, so they probably share some immune phenotype with the non-progressor children. However, to bring viremia down, they might also have a similar phenotype compared to the adult elite controller, or they might be using different mechanisms that we don't understand yet. So I want to show some preliminary data. It's quite complicated because this is a very small cohort. Most of them have rebounded, and most of them are also now on therapy. So in the last few months, we got access to 4 samples of individuals contronging viremias and still being ART naive, and I want to share those data with you. So here are the longitudinal data of one of the patients. So the first graph is viral load, and the second and the third absolute and percentage of CD4, and then CD4 and CD8 ratio. So looking at the 4 patients together, they all are females, they are all adolescents and coming from sub-Saharan Africa. They all have a good CD4 count and a fairly good CD4 and CD8 ratio. So we measured for the first one integrated HIV DNA, and it's quite low, it's 56. But in comparison to the last one that we have already measured, it's almost, this patient have integrated HIV DNA that is almost 1 log higher. So they are quite heterogeneous. We are still waiting for the result of the other 2 patients. And in the near future, we are planning to do some integration site analysis and proportion of replicative competent viruses. And to compare this group, we select a group of non-progressors, progressors, and exposed uninfected individuals, matched by age. And just to carry your attention for the progressors, we cannot say that they are rapid progressors because they survived for a long period without therapy, but at a certain time point they progress. So they have a CD4 below 350, but they are still, they had a slow progressor phenotype. So starting with HLA and KIR typing. So the most strongly single polymorphism associated with elite controller in adults is HLA-B57. As you can see in this adult data, having a protective HLA positively correlated with your CD4 count and negative correlated with your viral load. But when you add the pediatric data, we don't see the same difference. So for the pediatric HIV, HLA doesn't seem to play a role in disease progression. Looking at our 4 elite controllers, only 1 have a very protective HLA. And when we expand for other 3 that we also have access for HLA, only one other have a protective HLA. And also some of them has some HLA that are correlated with susceptible disease progression. So although their numbers are quite small to draw any conclusion about HLA, it's quite anecdotal to see that they are enriched with protective HLA. And looking at the KIR typing, I think there's a problem with this slide, but in blue are those KIRs that are presented. So KIR are more a receptor expressed by NK as part of a very complex mechanism of maturation and regulation. But there's been some association between KIR and some HLA combinations and disease progression. So we can see that all of them express KIR3DL1, which in combination with BW4 HLA alleles are correlated with slow disease progression and are enriched on adult elite controllers, especially those who are HLA-B57. But none of them express KIR3DS1, which is a KIR also associated with slow progression. And not all of them express KIR2DS4, a functional form, and this is also correlated with rapid progression. So for KIR, we have a kind of mixed effect, but again, it's the same for the HLA. The numbers are quite small and just like more for anecdotal than drawing any conclusion. So looking at the immune phenotype of the T cell compartment, we can see that the elite controllers and the non-progressive have a higher frequency of naive cells compared to the exposed uninfected, while the progressives have an increased number of transitional and effector memory cells. And when you look some activation and exhaustion markers, we can see that the 4 elite controllers have low expression of DER and PD1, while CCR5 is quite variable. And comparing with the other groups, we see that the non-progressor, but especially the elite controllers, have a very low expression of CD38 and HLA-DR, quite similar to the exposed uninfected, and a similar pattern is observed for PD1 expression. And looking at this heat map, so in red means low expression, and in green, so high expression. And we have here different markers of exhaustion and activation in different subsets. So we focus the attention here. We can see that the elite controllers are quite similar to the exposed and infected, especially for subsets that are more mature, while the progressors have a high expression of those markers. And interesting, the non-progressor lies between the 2 groups. And looking at immune activation and some correlations, so we see that co-expression of DR and 38 are positive correlated with viral load and negative correlated with absolute CD4. And we can see that the elite controllers in blue, they kindly cluster together. So this positive correlation with disease progression is also seen on PD1 and CD39. Looking at the CD8 compartment, we see a decrease towards a naive phenotype in the HIV-infected group and an increase on effector and terminal effector cells, which is what we expected of a chronic infection. And they also have more immune activation the HIV-infected group have more immune activation compared to the exposed uninfected. However, the elite controllers and the non-progressed have a less PD1 expression, so that although they have immune activation on CD8 compartment, they seem to be less exhausted. And again, we see a positive correlation with disease progression and PD1, and interesting, a protective effect of CD73, which is also observed in adult infection and on adult elite controllers. So looking some HIV-specific response, looking CD8 first. Here I'm showing the data of two, but it's quite similar for the other two. We can see a fairly good secretion of interferon gamma and degranulation upon stimulation with CAG, but we didn't see any difference between the three groups. And looking the other pools, we didn't see much difference as well. But the GAG-specific CD8 response on elite controllers seems to be more polyfunctional, expressing 2, 3, and 4 functions together compared to the other two groups. And when you look at granzyme B and perforin expression and co-expression, we didn't see much difference between the two groups, between the three groups. So looking at HIV-specific response, although as expected, the proportion of positive cells are lower compared to the CD8, we see a consistently higher secretion of interferon gamma on the 4 elite controllers compared to the other two groups. And it inversely correlated with viral load. And we didn't see a similar difference for the other markers. But again, we saw some more polyfunctionality on GAG-specific cells on the elite controllers. So for the NK phenotype, I don't have much data to show. We finished some ADCC and functional assays last week. But what I can tell you so far is that the progressor have expansion of CD56 negative, CD16 positive, what is also observed in viremic adults. And the progressor and the non-progressor have a preserved CD56 gene and CD56 sprite. So although we cannot draw any conclusion from here, we can say that they are probably less dysfunctional on the non-progressors and on the elite controllers. But we still need to look at the data. So in summary, we can say that the elite controllers in the pediatric population, they existed, and they are strongly biased towards female sex. Viremia control are usually short-lived in this population, although you have some individuals who has been controlling viremia for a long period, and they usually start years after infections, quite different from the adults. We didn't see any particularly enrichment on protective HLAs, and also some cure and HLA combinations are quite mixed. We did find a very low immune activation on the CD4 T cell compartment, and we found some GAG-specific response with a higher production of interferon gamma that negatively correlated with viral load. We also found CD8 T cells less exhausted and more polyfunctional compared to the other two groups. And the NK cell, we cannot draw much conclusion, but we can say that they probably have a less dysfunctional immune phenotype. So in children, because of this tolerogenic immune system during early infancy, they rapidly progress in disease, but around 10% of them maintain a normal CD4 count. And on the way, some of these non-progressors, they progress, and some of them achieve adulthood with a normal CD4 count, so we call them long-term non-progressors. And a very rare group of individuals become also an elite controllers. And we can This viremia control is probably polyfunctional, and we just started working with these patients, but we can say that it seems that the HIV-specific response are more polyfunctional in these individuals, and they have a more cytotoxic CD4 T cell. They probably have a more functional NK response, and we still need to look on the viral factors of these individuals. So if you think remission has a treatment-free condition, we can say that the adult elite controllers are a good model for remissions, as they have a very long-term control of plasma viremia, and they maintain on average normal CD4 count. However, if you consider remission as a disease-free condition, we have to remember that most of the elite controllers have a moderate immune activation and mucosal damage, and increased frequency of non-AIDS-related comorbidities. For pediatrics, you cannot say yet, but what we found so far is a very low immune activation on the CD4 compartment and an absence of enrichment of protective HLA. So although they have a more polyfunctional CD8 response, this might not be the major driver of viremia control in this population. So I want to stop here, and this is still an ongoing work, and I'm happy to hear your suggestions. So I want to thank my supervisor, Professor Philip Guder, and my co-supervisor, Professor John Frater, and my group who is helping. I'm more a clinician, so the group is quite helping me with the lab work. I also want to thank you, our team in Durban, South Africa. So this is a picture of our clinic in Durban. And these patients come from different cohorts around the globe. So we have collaborations in my country, in Brazil, in other groups in South Africa, here in Spain, Thailand, and other groups in the UK and also in the US.
Thank you. Thank you. So we have 5 minutes for questions.
Thank you, Vinicius, for this nice talk. I have 2 questions. The first one, have you tried to do inhibitory inhibition of viral infection in vitro with NK cells, for example, for these children?
Yeah, we just finished the ADCC and the functional assay using K562 and other assays looking at the NK functionality, but we will analyze the data and see what we can do next with, like using some inhibitory assays as well.
Okay. Yeah. And the second question is a little bit surprising, the lack of the low representation of protective HLA.
Yeah.
27, 57. So one possibility is that if this protective allele is coming from the mother, then that escape mutants of the virus have been generated.
Yeah.
The mother and presented to that was described by Bruce Walker a long time ago. Do you think that could be one mechanism to explain why there is maybe the question if you have to look at the escape mutants against B27 and B57
yeah. Yeah, we don't have access to all mothers' HLA to see which one, but we are planning to do the full-length sequencing of those viruses. It's interesting to know that most of them just start controlling viremia by the age of 6, though they have like viral replication during like a long period of infection. So they might be a mechanism, but it's interesting to know that they only start controlling the virus after like years of ongoing replication.
Hi, how are you? Adriano from Mesa. Congratulations for the study. I have a couple of questions. The first one, it's very interesting how it takes so long for these children to develop the control. And I was thinking, something that makes different kids in terms of T-cell responses is that the whole T-cell repertoire is being formed during the first years of life in terms of immune function. So do you think that somehow there could be a selection for maybe conserve epitopes in these kids, and this is how this mechanism could be contributing to the control? That would be the first. And the second question is looking at the other side. Do you think there's any differences in terms of antigen presentation? So the mild compartment, and did you see any differences in terms of the ribs and response sites in this?
Yeah. So for the innate response, we didn't have the opportunity to look yet, especially because this is more like historical samples, and like usually like to look like dendritic cells, it's better to have freshy samples, and they come from different cohorts. So we didn't look yet, but we are planning to do some single-cell RNA-seq, and it might be a good way to look some subpopulations. And for the cytotoxic response, yeah, during these, along the years, they become they develop a stronger type 1 response. So they probably increasing their capacity to be more cytotoxic, these cells. But because they are not particularly enriched with those HLAs that target specific regions of the virus, I'm not sure if although they seem to be more polyfunctional and specific to GAG, I don't think they are the major drive of viremia control here.
But maybe, TCR sequencing could be very interesting.
Yeah. Thank you.
Cecilio Rodríguez from Instituto de Salud Carlos III. Thank you very much for the talk. I am wondering, perhaps these pediatric controllers are more related to the Visconti people. I'm thinking in the HLAs. It looks like they are more similar to the HLA of the Visconti people. No?
Yeah, like the Viscon, they are not particularly enriched with the protective HLA, so in that sense they are quite similar. That's why we are also quite interested to look at the NK cells, because there are some correlations with remission on this cohort and the NK response.
I just have a question. You just said, can you hear me? Oh. Okay, you are saying that in this group of patients, CTLs may not be controlling the virus. Okay, so what about B-cell response? You haven't said anything. So are you planning to study humoral response? Yeah. I know that I'm not in the correct audience, but I work with humoral response. And
okay. We didn't touch yet on the B lymphocytes. We did Western blot, and they all have, as expected, strong bands. But it's in our plans to look at the B-cell response. But if you look at the pediatric data, usually when we measure broadly neutralizing antibody, it's in children correlated with disease progression. So the non-progressor have a lower proportion of broadly neutralizing antibody compared to the progressor children. So that's,
but this is a special group of patients.
Yeah,
Maybe
exactly.
you can look for FC functions or differences in the B-cell compartment.
Yeah, sure.
Okay, one more question. Vinicius, one interesting finding is the low immune activation observed in the pediatric elite control population compared to the adult elite control population. Even those children that have ongoing replication have low immune activation. So, do you think there is a role for this low immune activation for interferon alpha? Have you explored the interferon alpha route, the type 1 interferons?
No, we didn't.
And the role of this?
No.
Because this is something that is interesting, and interferon alpha has been proposed as the modulator of immune activation in adult population.
Yeah, it's interesting, yeah.
Okay.
That's only quick. Have you taken a look to thymic function in this?
Say again?
Thymic function, the thymus?
Oh, no, we didn't.
Because I think it could be important that this subject has got an extraordinary thymic function.
Yeah, no, we didn't. Actually, we only got access to those samples 4 months ago, so we were trying to, because they haven't been described in the literature, we are beginning from we're starting from the beginning, so from now on, according to the results, we are guiding, because we don't have many samples available, and most of them are now on ART, so we need to be quite criterious while we are looking on these samples.
Thank you.